Yep, here we go again. Hi. Hi, hello, and welcome. Microbehunter here. Yeah, welcome to another Saturday live stream, to another Saturday microscopy live stream. Yeah, today I'd like to talk a little bit more about microscopy hardware. Don't worry, not only, we'll be looking at several specimens as well. Yeah, and I'm just checking again if everything is working here. Sound is okay. Thank you very much. Let me do some other housekeeping here. Let's see if this works. Yeah, we've got 16 people currently online. More are joining in. Okay, so here we are again. It's always a little surprise to me as well how quickly a week goes by. Christmas is slowly coming again. Is usually the time when people start doing some shopping for Christmas and maybe also some microscopes. And because I've got a pretty decent, also slightly expensive microscope, I decided today I'd like to go a different way. I'd like to do a little bit of microscopy with fairly cheap microscopes. And basically, this is where we are. Not only that, I would like to also show you some of the issues or features or problems or things that those cheap microscopes have. I just want to say again that I've got a couple of them here. I'll just show them to you. I'm also going to show you some of the problems of upgrading a microscope. I've got some objectives here, and unfortunately, even though they fit, size-wise, I'm not able to put them on here. I'll explain to you why. As always, if you have any questions, please feel free to ask them in the chat box on the side. If the questions are on topic, of course, then I'll try to answer them right away. If they're off topic, then after approximately 1 hour, I would like to then answer them collectively. I've seen that was already the very first question is about rubbing alcohol and whether it can be used for dehydration and so on. Yeah, so I'll talk about this as well. If you have any specific questions specifically for me, please indicate this by indicating this is at Oliver or at Microbehunter, so it's easier to find the questions. Yeah, hello from Northern Germany, from Lebanon, from the UK, from Germany, Belgium, England, USA. Okay, and so all around the world. And what I'm going to do is the following. I'd like to give you a short intro into this microscope. I just want to say this is not a sponsored video here. A couple of years ago, I did receive this microscope free of charge from a company. And I did make a video of that already. So you can find this in my other channel. It was kind of a review video. But today, this is basically, we're completely independent of that. So I also would like to show you what I've done here. I'm going to flip my camera up. Here you see my monitor. Okay, and here, look, this is what I've connected here instead of an eyepiece. So this is essentially where you'll also be able to see the image through this microscope here. Okay, so that's essentially also something that I'll be showing you. And I would like to talk in the first couple of minutes a little bit about theory, so to say, because I do have a second one over here as well. Look, okay, and those microscopes, they're fairly cheap. They cost around, I don't know, maybe around 80 to 100 euros or so. And essentially, the objectives here is something that I'd like to have is a closer look here. Okay, so first of all, because those two microscopes that I have here are a little bit, they're quite similar on the outside, but the objectives are of a different size, and I would like to talk about this. And where should I start? Who cares? I'm just going to start right away at the objectives here because I would like to show you something. Because one of the earliest, immediately when people buy a microscope, sometimes people like to start upgrading them, and I just wanted to show you now something interesting. It's not that interesting, but simply something where you have to be a little bit aware of. So this is a fairly low-cost 10 times objective, and you see over here 160 millimeters, and I don't know if you're able to see it over here. This also has 160 millimeters. I'm going to take this off here. Just a second. I'm going to take this one off here. This is the 10 times, if I'm correctly informed as well. And let's have a look at those. This one, this cap, I also have to remove. Let's have a look here. And look, isn't this interesting? They have exactly the same size. Right. And the length is a little bit different, yeah, but otherwise, wow. This seems to be quite promising here. So maybe I'm able to upgrade the objectives here. I just want to show this to you. The other one that I have, right? I'm going to take out that objective as well right now. And let's see if I can even unscrew it. Yes. Let's have a look at the other objective. And here it is. And if you just put them next to each other, then you're going to see that the other microscope, has a different diameter. So not all of those low-cost microscopes actually have the same standard. So this diameter here is significantly smaller. So you're not able to put this objective on this microscope and vice versa, even though the two microscopes actually look extremely similar on the outside. So that was an interesting observation. It's not a big issue or problem or anything. It just kind of shows a little bit that from the outside, even though the objectives here have the same size, they're not interchangeable. But these, and they're also significantly smaller than those standard size objectives. But this looks kind of a little bit promising here. So maybe I can upgrade the microscope here, because this microscope here has magnifications 4 times, 10 times, and 40 times. So maybe I can, even add a 60 times objective here. And indeed, look, I can maybe flip this over a little bit. The camera is a little too heavy. Indeed, I can. I'm indeed connected, but there is an issue. And the issue is as follows. And you're probably already able to see that. Even if I move it all the way down here. It's still, the problem is it's still too large, and I'm still not able to get a clear image. Okay, so this is a little bit the problem, that I'm not able to lower it yet further. I tried it out. It's a little bit of distance is missing here, and over here, you see that this part here bumps the base here, and therefore I cannot lower it any further. So this means that even though it is theoretically possible to exchange the objectives of those microscopes here, for all practical, it doesn't work. Okay, because and that's now basically something where I have to explain this to you. The so-called the conjugate distance is different, because in the 160 with the 160 millimeter objectives, there are again two different versions. If you just put this next to each other, and you're able to see that it says here 160 over here, right, and 160 over here, and this basically means that the size over here is the same. However, the barrel length is different. So these are so-called short barrel objectives, and they have a so-called conjugate distance of 185 millimeters, and they have a conjugate distance of 195 millimeters. So that shows already a limitation a little bit, that, unfortunately, not all of these objectives are actually interchangeable. So this is something that you might want to take care of a little bit, that, microscopes are not always easily upgradable because of that. So I'm going to, every now and then, I'm going to check over here. Oliver, can you show how to remove that black metal ring on the objective? Are you referring to this here? This black metal ring, this is actually not metal. Okay, this is actually rubber. It's a decoration and simply glued on here. I'm not going to remove it now, but actually, you see, if I think it's almost like a rubber band, if this is what you're actually referring to. So and there's also a question of color coding, and indeed there is color coding here. So yellow is 10 times, red is 4 times, and then you've got 40 times is blue, and usually 100 times is white. So you can actually see simply by looking at the color, you can actually know which objective it is, because sometimes the labeling is not readable. Okay, so what I'll do is I'll just put this back here, but I just wanted to show this to you, that the whole issue with objective standards is a little bit unfortunate here, and also if you have so-called infinity objectives, these are pretty large objectives from modern microscopes, then of course they're not exchangeable either. Okay, so I'm just every now and then I'm just skimming a little bit the questions here. Okay. So there's a question basically about denatured alcohol and distilled water. I'm just going to answer it right away. Can you use denatured alcohol for dehydrating specimens? Of course you can. However, denatured alcohol is just regular ethanol with which contains an additive so that you cannot drink it. So it is free of alcohol tax. That's why it's called denatured alcohol, and otherwise you could be able to drink it, and then people would simply drink it, right? And if you want to have it, and usually some countries put alcohol tax on it, but in the case of denatured alcohol, they added a substance which makes it bitter or not drinkable, and then it's free of alcohol tax. So absolutely no problem, because for removing the water from specimens, you would just use this alcohol, and it's going to be fine. Yeah. And the question is about distilled water. It doesn't matter if you use distilled water or tap water, because as soon as you add distilled water to a specimen, it's not distilled anyway anymore, because all of the impurities and minerals of the specimens are going to go into the distilled water anyway. So it doesn't really make a difference. So distilled water and pure alcohol, non-denatured alcohol, are generally used in the lab if you're doing highly reproducible experiments. But just for microscopy, for dehydrating, it doesn't really matter. Isopropanol is more expensive and does not mix quite as well with water, and therefore what they often do is you use isopropanol or isopropyl alcohol as a last dehydration step for preparing specimens. Okay, so that's the one thing I wanted to show you. There is another thing I want to show you, and because I cannot flip this microscope over now, I just want to show you something which is a little bit unfortunate, I think. And you have it with all of those cheaper microscopes, and this is something that I do not understand why they do that. So I just want to show you. I'm going to flip this around now. Yeah, and that is this ring down here. This is usually a plastic ring here that you can rotate. This is filter ring. In this case, you only have holes of different sizes. In some microscope brands, they put some colored filters in there. Yeah, but the question is, what's the whole point of this? It's completely useless. Because it does not act like changing it does not act like a diaphragm, because the hole diameter is way too large. Yeah, that's totally pointless. Yeah, so there is zero. I've seen zero effect, but it's still possible. What you can do is you can actually put a little, a small piece of a credit card over it with a tiny hole, and that would actually then have an effect of not only reducing the light but also increasing the depth of field. Yeah, so that's a little bit. I think a feature here, which is not a feature because it really doesn't fulfill as such any function, right? So those holes are way too big and they don't have an effect. And so what I've also tried to do is I tried to include over here some polarizing filters. I thought, wow, cool, I can use different polarizing filters now. However, there is another problem that appears, and that is that the lens that you have here in front of on top of the stage destroys the polarization again, because sometimes these are plastic lenses. Yeah, so that's also a little bit of a pity. So what you'd have to do is you'd have to take out this plastic lens, and then it's probably going to work again. Yeah. So I just wanted to show this to you. And there is another thing that I'm going to demonstrate now, but I think we're just going to have a look at a couple of specimens first. And I'm going to put this here. I don't know. Yeah, and I'm going to switch over to the microscope view, and now I'm not going to use my later on I'm also going to use my big microscope, okay, but right now I'm going to switch this on. Yes, we've got some light here, and I'm going to use my some of my antique specimens that I've bought. I'm still looking for some nice antique specimens. I'd like to find a couple of nice diatom slides. Have not ordered them yet. I promised that I'm going to do that, and I will do that. But I just do not want to buy any slides. I'd rather like to buy nice-looking ones. Okay. So and now, okay, this is now on here, and now I have to find it. And this is already the first sign that how difficult it is. And here it is. And this is the cross section of the spine of a sea urchin. If you watched some of my previous videos, then this is going to be a familiar one. However, it is overexposed, and for this reason, I would like to show you. I would like to yeah, simply go down a little bit with the exposure. I'm not going to use all the exposure, but I'm going to now manually adjust it. Okay, yeah, and here we go. And this is now using the low power 4 times, and I think it looks quite okay. Yeah, but there is something else that I would like to show you then later. Okay, so this looks kind of quite okay. Let's move up a little bit with the magnification. Okay, you can readjust the focus. I have to go up a little bit with the light as well, if I'm not already at the maximum. Okay, if I need to make it brighter, then I'm going to go again into the control, the video control, and I need to go up with the exposure time a little bit. Yeah. So, okay, here we go. And you see, with 10 times and 4 times, it looks quite okay, actually, I think. I also have to say that the camera itself only takes basically magnifies a small section from the center. So it depends a little bit also on your camera setup. Okay, and let's have now a look at the 40 times. Okay, and this is where things already start to become a little bit more interesting, because we can see that it's already significantly darker. It's significantly darker. I have to always find a little bit the one where I can, and I'm going to go up with, and you see it responds very slowly because of the slow frame rate. So you see that it's also with a video system and the generally low light, it's not suitable very much for video, but for static images it's fine. And that is now basically the cross section of the sea urchin using the highest magnification here, and it's decent. Okay, it's fine, not super crisp. But this also has to do a little bit with the fact that the camera magnifies so much as well. Yeah, so there's a lot of some empty magnification as well. Yeah, so this is actually might be okay, but there is an interesting thing here, and that's something that I also just discovered a little bit by accident. And that is that normally when you get a picture from under the microscope, the image is inverted. Okay, so it's like turned upside down. In other words, not flipped, but actually rotated 180 degrees. That's really not a problem. Everyone knows that. It doesn't disturb, right? But the problem but the interesting thing that we have got with this microscope here is that in here. Let me show you again where is this one here? Yeah, in here, up here. Yeah, there is a mirror. And what this mirror, to redirect the light from the bottom into the tube, and what this mirror does is it doesn't rotate the image, but it flips it vertically. Yeah, so that is a little bit of you don't notice that, you don't know that because unless because the specimen is so small, so you don't see that. But that is something where that you just have to be aware of. The image that is therefore produced by these cheap microscopes is actually not the way that it looks in real life, because it is flipped, mirrored on one axis. Yeah? It's not a huge issue. You just, if you want to take a picture, you just can flip it again back to the original. But still, it's something that one should be aware of. Okay. Yes, there are now a couple of questions. So, yes, Oliver, I recently saw your cheap Telmuscope review. My friend bought that same scope for only $25 on eBay a couple of months ago, it's very serviceable for that price. Yeah, all of those cheap microscopes. The Telmo microscope is basically the same in the same category as those here. All of those cheap microscopes, if you look at them, interestingly, they use a lot of the same parts. I would not call them toy microscopes, though. Okay, the reason is because yeah, for example, this one is made of metal, right? They're suitable for children, sure, and I think also anyone who is generally interested a little bit in starting off with microscopy and who does not want to spend a lot of money, that's fine. But of course, those microscopes are a little bit limited in because they don't have a condenser and so on. But I think the real, how do you say, under quotation marks, limitation is that those microscopes, due to the small size, are not really suitable for long-term observation. Like, for example, if you're really sitting many hours behind the microscope, you probably want to have binoculars, you want to have a proper size of the microscope, you want to have adjustable eyepieces, all these things, right? So there is a lot of ergonomics that goes into the high-end microscopes, because these microscopes are sold for research institutes and hospitals, and sometimes those folks are actually sitting behind the microscopes for hours and hours and hours every day. And for this reason, they're placing a much stronger focus on ergonomics, and it's not necessarily primarily image quality that's the issue. Okay, just wanted to just wanted to say that. Yeah, there's also a comment here. Agreed, the toy scopes I've seen seem to have plastic objective lenses. Yes, and the objective lenses are significantly smaller, and honestly, they're very difficult to use. Yeah, so those microscopes here, I even can use them. You can even use them as field microscopes and carry along and so on. And if you actually get those, then you are going to see that they also come packaged fairly seriously, usually in a very boring looking cardboard box. Yeah, basically like bigger microscopes. Another thing, I just want to show you a little bit of what I do consider, and then we can look at a couple of more of these specimens here. What I do consider a little bit, how do you say, annoying is not annoying, but I mean, you can press here on the back, and then you can fix, basically, connect the slide here, clamp down the slide. But this, of course, also limits the movement a little bit. So usually what I do is I kind of just move it around like this and just let it stand. Yeah. So that's basically Let's have a look at a few other specimens here. I mean, if you watched a couple of my previous streams, then you've already seen some of those. I do also want to make a short announcement now. Let me quickly check how many people are watching now, because next Saturday, there's 60. Okay, that's quite decent. 60 people have joined around. I just want to say that next Saturday, for private reasons, I will not do a live stream. Okay, and what I'll do instead is I will release a video at that time, probably as a so-called premiere. This means that it will be pre-recorded, maybe not quite as long as those live streams here, but if you want to, you can still join in and use the chat function. Okay, so, and what I would like to do is I'm going to also show you a couple of slides. So this one over here, let me have a look. I'd like to actually look at a couple of insects again. Maybe, where are they? There are a couple of very nice specimens here. That's a plant cross-section that what I have here. Let me see, what is this? Yeah. Okay, that's another one. Let me see where I've got some up. Those slides here, by the way, are, I can show them to you also over here. Those slides here are antique slides. Okay, they're probably around 100 years old, which I bought. So yeah, they're very high quality slides, and some of them were even commercially made, obviously, and sold. And we're just going to put those slides here now also on here. We need to. Our lamp is active, and let's have another look if this actually works. Okay, I did not read the label now, but one of the problems with this camera setup is that it does it does magnify a little too much. Okay, I have to. What does it say? It's a beetle. It's a larva of a beetle. Okay, yeah. And let's have a look if we're able to look at the mouth parts in a little bit more here. So here we go. You can actually see, honestly, I tell you why I'm, okay, it's a little bit reddish, the color. It's a camera thing. One of the things why I'm so fascinated by those cheap microscopes as well is because back in the day, when I was still studying at university, they didn't have those. This was also now many years ago. And if you wanted to get a microscope, you actually had to buy a fairly expensive one. So my first microscope that I bought was an Olympus CH40. Obviously not manufactured anymore. I paid a fortune for that. At that time, I didn't earn any money yet, so I had to save a lot of money together to be able to buy that. And I used that microscope for a very long time. Still have it. But honestly, those cheap microscopes for a fraction of the price, yeah, are also able to do their job. Yeah, not quite as conveniently maybe as the larger microscopes, but yeah, it also kind of works. Yeah. So this is simply something where I would like to advise those people. Usually, when they ask me which microscope should I buy, that's too difficult to answer. I mean, it depends what you want to do. But if you're just interested, yeah, buy yourself a cheap one first, try it out. Yeah, and then you can always upgrade later. Yeah. So I'm going to again pause a little bit here and. Yep. Yeah, okay. I'm following you also now on Blue Sky. Keep the interesting stuff coming. Yeah, because you're asking, what I've done is the following. There is this hype now about this new social media app called Blue Sky, which has been exploding in the last couple of weeks, exploding in the sense of user number. And I've not been on these yeah, on Twitter, and I have a TikTok channel which I'm not using a lot. I tried to publish some pictures on Instagram, but yeah, but then basically I was not happy with those platforms because the things that I posted they were not always shown to the users, and so I tried out now this new app called Blue Sky, which is in the media quite a lot these days, hyped a little bit. And I have to say, I'm positively surprised by that because of its simple use and its straightforward and clean interface. And so what I'm going to do is I'm also simply going to post links to my videos and pictures maybe also on there. So if you want to follow me there, I actually made a short, how do you call this, a short cut as well. So I'm going to quickly go over here again. Yeah, so this is basically the handle. So microbehunter.bsky.social. And if this is too complicated, the bsky.social, then I've also made a redirect, bsky.microbehunter.com. Yeah, so that's basically a subdomain from my webpage. And then you're going to be redirected to there. And simply what I do is I simply cross-post links and pictures and things like this there as well. We're going to see how this works. Yeah, and if this social media site will actually be successful in the future or not. We're going to see. I'm just going to play around a little bit with it. And yeah. So that's, look at all of those. That's the exoskeleton here. Yeah. Yeah, quite nice. So another one that I would like to show you. So that was the beetle. What does it say here? The larva of a. I have a problem reading the old handwriting, but that is in any case. And what is this here? Ah, yeah. And another issue that I'm having is that I'm starting to have vision problems already. So I need to put on my glasses that I'm able even to read this here. Okay. Tongue of a honeybee. Oh, okay. Yeah, that's also one here. That's the tongue of a honeybee. And you see that every time the focus is lost, and the reason is not a problem with the microscope. The reason why the focus is lost is because the slides have a different thickness. Yeah? And because at that time the slides were evidently not quite as standardized. So that here is the tongue of a honeybee, and look, really nice slide preparations. So what I'm going to do, just for the fun of it a little bit, let's have a look at this also under my other microscope, simply so that you see a little bit the field of view difference of the camera system. Yeah. So basically, you see that there's this blue background here. Yeah. So because I did not stretch the image, I just basically took the whole image as it is from the camera, so that you're able to see the full image, right? That's the reason why it doesn't cover the full background. And let's have a look now how this actually looks. Where is this? How this looks under my other microscope. So I have to put in, yeah, so this one here is now at 10 times. So I'm also now going to use the 10 times over here. Okay, so let's remove the arrow because it disturbs a little bit. You see that I currently have the 4 times here. You can flip out if you check, you can flip at the top, you can flip out the condenser to give it a better field of view. I'm going to remove the prism. It's 4 times, yeah. And this here is now 10 times. Let's have a little comparison a little bit concerning the, yeah, image clarity and sharpness. You see, this is how it looks like using my other microscope. I mean, it does look a little bit, definitely more crisp and better resolution, but I think the small microscope also does its job. Yeah. And so that's basically, I'm always still fascinated by. Okay, I'll tell you what fascinates me about these things. It's not only the slide preparation, or I've got more a biologist's type of fascination here, and that is the complexity of anatomy that we have here, and that's the tongue of a bee, right? Where in the world are all of these details stored? Well, they're stored, of course, in the DNA of the cells. But the question of how anatomy is encoded in the DNA is something really fascinating, because you're not able to locate any place on the DNA which actually describes how it's supposed to look like. Yeah, it's we biologists call this so-called emergent properties. So the question of how anatomy is encoded in DNA is still, to a certain extent, a mystery. How complexity is able to appear. So in other words, to put it in simple words, organisms are way more complex, than what you have on the DNA. So complexity appears, during the development, and that's something difficult for me to a little bit to explain. But to use an analogy, there are mathematical formulas which are able to make so-called fractals. These are very, very intricate, beautiful, pictures, those fractals. But the mathematical formula is very simple. Yeah, so complexity is able to appear in the form of emergence, as it's called, by having the different parts of a biological system interacting with each other. I don't know. I'm getting carried away a little bit, but that's simply something that I'm kind of interested in here. Yeah. So. Yes. Okay. There's a question about paramecia culture from Paul. I will talk about this later on, because I do want to share you some experiences, because I was semi-successful, and I can indeed pass on some suggestions of what you can do to get a paramecium culture, because many of you probably already know I'm a teacher in school, and we are, of course, also doing microscopy in school, and we also need to grow our paramecia for the students, so that they have something to look at under the microscope. And this can be a little bit frustrating, because not always will the paramecia grow. And then they grow quite nicely, and then all of a sudden they're gone. And why are they gone? Well, because there is a natural progression, and I'm just going to give you some tips later on what you can do to make this a little more successful. Okay, so let's have a look at this one here. What is this? I have to find the. I have to find the. I'm going to go down with the magnification, of course. I know what this is. Yeah. So again, here we are right now again with the looking at this with the introductory with a cheap microscope. This what you see here is the exoskeleton of an insect, of a so-called of a blowfly. Okay, it's known this as this is so-called this spherical. What is that? Insects do not have lungs, but they still need oxygen. So how do they get oxygen? Well, this is basically the opening in the exoskeleton, in the outside part, where so-called the trachea. Tracheids are air tubes that go from the outside into the body of the insect. These are tube-like structures that kind of branch off from the outside, from this opening into the insect. This is then these are the canals how oxygen is then supplied to the cells. Yeah? So it's basically this here is now the entry point for air. Yeah? And this is where air flows in and out, and you can see that, and that's quite interesting here as well. And it's now what I'm guessing here. Great. I have no arrow over here, unfortunately. Yeah, I don't have an arrow. I forgot to include my arrow. No, it is actually here. Where is it? I get it. Just a second. Where's the arrow? Why can I not see the arrow right now? Because I have to put the arrow all the way to the top. Here it is. Oh, okay, here we go. So you see all of those little things in here. They seem to be covering a little bit this spherical, and I think this might be a little bit of a protection against dust and dirt entering the whole system here. I just wanted to show it to you. And again, we're going to put it again under my other microscope. Yeah, and we're going to see how this actually looks like. And I always have to switch back and forth and refocus. And this is how it looks like. And actually, you do see that the resolution is quite a bit better. It's also the same magnification here. You see more details. It looks a little more contrasty. Of course, what do you expect? Yeah, but still, I think. Yeah, now at the next higher magnification. Yeah. And of course, we also have a condenser that we can use. Yeah. But still, yeah. So let me see. I know that arachnids, these are spiders, have book lungs, and crustaceans have kinds of gills. What about millipedes? Good, interesting. Millipedes and centipedes, also arthropods. Would be interesting to find out. Maybe some of you can research this. Maybe some of you can research this. Do millipedes and centipedes, do they also have spiracles and a tracheid system? Okay. So a question now over here. What microscope are you using now? Well, the microscope that I'm using now is not my CH40. Okay, this one is also a BX53. It's what they call, when I bought this, the guy who sold it to me from the company Olympus, he said it's actually a very common microscope, also used by doctors. Yeah, and the doctors, he said, have to sit behind the microscope for a very long time. He said if people sit behind the microscope 8 hours a day, pathologists who basically their job is to analyze the slides that basically come in. So let's say they're taking maybe some kind of a biopsy from a patient, and they're making the slides, and then it has to be looked at. Those folks who are doing this are basically sitting, I don't know, many hours a day behind the microscope and just processing slides, looking at slides and then documenting that and then giving a diagnosis. And for that, of course, you need a microscope that is very ergonomic. So, for example, my microscope here, you can adjust the height of the eyepieces. Yeah, you can flip it up and down. Of course, the two eyepieces, you can adjust the width. There is for controlling the slide. It goes all the way down, so I can rest my hand on the table. I don't have to hold my hand in the air to operate the slide and the focus knobs, because they're deliberately so low that I can rest my hand on the table. So there are a whole bunch of ergonomical features, right, that this microscope has. Right? So that's why they're designed already with long-term use in mind. So that's I just wanted to show it to you. Concerning the cost, I'm not going to talk about cost so much, but these microscopes are modular in design, and this means that how the actual cost is determined primarily by the parts that you put on it. So basically, you cannot go to the company and say, okay, send me that microscope. They're going to say, we can't. What do you want, right? We've got several eyepieces for selection. We've got a whole range of different objectives to be selected. We can have different condensers that you can connect. Probably differences in LED or halogen. What do you want? Yeah. So we're going to put the system together for you, right? So these microscopes are not sold out of the box, but you actually have to get in contact with the company, and you got to tell the salesperson what you want, right? And then the parts are going to be ordered. And I remember when this microscope that I have right now came not in one box, but in two big boxes. And in each box were, I don't know, 10, 20, 30 smaller boxes, because every objective was packaged separately, every eyepiece, everything was separate, and you had to assemble it. Who had to assemble it? Well, I did it myself. But when it arrived, the company salesperson said, let's make an appointment so that I can put the microscope together for you. It's part of this package, right? I said, no need, I already did it myself. I just want to show you with that it's entirely different how this actually works than when with the out-of-the-box microscopes that you normally would buy. Yeah. So Wikipedia, millipedes breathe through two pairs of. They also have spiracles. Thank you. Located ventrally on each segment, nearly the base of the legs. Oh, okay. So they have multiple of these spiracles. So in each segment, each opens into an internal pouch and connects to the systems of tracheae. How do you pronounce this? Okay. Yeah. So basically, interesting. So they have multiple each segment. So let's move on, because there are a couple of. Let's check the time. I'd like to, because you might want to look. We might want to look at a few more. I'm going to show you now one of my. I'm going to make, by the way, I intend to make a video on my favorite ones, and I do have a favorite one over here, and I did make videos already before a couple of months ago. But people don't like these videos, and I tell you why. Because some people have certain concerns here, and I want to show this to you. But I consider this one to be a very interesting one, but not one that is very, how do you call this? Very nice. Are you able to read this? Here, let me refocus this here. Okay, this is also an antique slide here. A section of the finger of a fetus. 4 months old. So that's basically the finger of an unborn baby. So, and here it is. It's stained. I made a video of this, and I think it's quite fascinating. It's not a very nice looking slide, so definitely not as beautiful, by far not as some of the plant cross sections that I've got, or some of the cross sections of the spines of the sea urchin that we just looked at. But I think just from a specimen standpoint, it's quite interesting. So let's have a look at this here as well, and let's put this in here. Okay, so this is basically just histology, what we're doing right now here. I unfortunately don't know what stain they used. This seems to be the fingernail over here. And over these, that's very typical. That's most likely a blood vessel over here, and maybe some red blood cells. The reason why I think it's a blood vessel is because you can actually see over here, yeah, the opening of the blood vessel over here, and blood usually looks like this when it's coagulated, yeah. The individual red blood cells are clumped together. You're not able to see those. Yeah? So, yeah, and yeah, these here. I wonder if these could be. I think it could be either collagen fibers, yeah, of the tendons, yeah, or muscles. The image quality is miserable, but I do not attribute this to the microscope. I attribute this more to the camera. Yeah, these USB cameras, and that's not the newest one. Those USB cameras don't have a very high dynamic range. So this is also quite nice to see. What you have over here, these seem to be some kind of, if you zoom in a little bit, some kind of like glands that release, maybe sweat glands or so, that open up to the outside of the skin. Yeah. So, but again, let's have a look how this compares to my other microscope. So, I'll just put this in here again. So, by the way, same magnification. And here we go again. Yep. And we do see more details, obviously, but yeah. I wonder. Yeah, I had some. I tried to identify some of the parts. Not always easy to do that. I'm not so much into human anatomy, but I was able to identify a few things. And what I'm going to do now is just want to show you here. Yeah, this here is seems to be the fingernail. Yeah, over here, those fiber-like structures could be either muscles. This brown thing here, spot, is probably coagulated blood in the blood vessel. Yeah. And you might wonder, why is it so round here in the back? And the reason is because when you cut a specimen diagonally across, yeah, then basically it will also look round, yeah, here on the back. Yeah. So that is one of the more, I would say one of the more unique specimens that I have. I do also have another unique specimen that I would like to share with you, and which I consider more interesting from the, how do you say, from a biological viewpoint. And I have to find it. That's also one of my, where is this? No, it's an elm tree. That's an elm tree. Let me just. I have to go through all of those slides here. If I only find it. Let me see. No. I mean, I've got so many of these now that I have a problem actually finding it. Let me see, otherwise I'll just show you another one. It's okay. No. I actually did sort it and now I don't know where I put it. Yes, I found it. Here it is. So that's another unique one, rather unique one over here. Let me show you again here. Let me see the desk view here again. Look, yeah, this is a tail of a monkey as a cross section. Unfortunately, I'd like to know which monkey it is. I'd like to know when the slide was prepared. I don't know what this means. Yeah, but it's also an antique slide, probably around 100 years old. And yeah, also very nice specimen. Let's have a look at this as well. And the 5 minutes here we go. Yeah, and this is actually a nice. This one here in the center, for example, very typical. That's the bone. Yeah, this is bone. Yeah, definitely. And this is the bone marrow in here. We can see. Yeah, yeah. over here on the side. Where is this? Or here on the top, muscle cross sections. Yeah, over here for basically there's one at the top, one on the bottom, left and right for moving the the tail back and forth. What do you see over here? Those brown structures here are hair follicles for the fur, extending outwards. You see occasionally some blood vessels as well. This here, for example, is an artery. Why? How do I know it's an artery, not a vein? That's because of the thickness of the wall. Arteries carry blood with high blood pressure, therefore the wall has to be thicker. And so that's an artery here, carries blood away from the heart. So there are quite. So this is actually a very nice specimen over here, because you can see several of the things here, including muscles, blood vessels, bone in the center. Again here the bone. If you actually go, let's try this. Let's go up a little bit with the magnification. If we're able to see the osteocysts, these are the bone cells, those tiny dots here, because bone is a living structure, and those bone cells, they deposit calcium, in the bone, making the bone hard. So quite a nice specimen in my view. Let me have a look here. So far there do not seem to be a lot of comments or questions. That's an interesting one. There's this comment here. I spent probably an hour talking with coworkers about the moral and ethical factors around the creation of the slide. Got a lot of conversation. So this is actually I also got emails concerning moral ethical considerations, and specifically not necessarily concerning animal rights. But actually, I've got several slides containing human specimens, right? So, and I did receive some time ago an email from someone who ordered himself a slide box, and then realized by reading the labels that actually, well, these are human specimens, and he was asking me then, this is about the ethical considerations. I mean, did the people actually give their consent to be used? And I say, of course one can assume that, because in the field of medicine, these specimens are extensively used, right? So there is no, how do you say, no lack of also in medical for dissections and so on. There is certainly, I think, no lack of material available, right? And you don't need a lot of material to make many slides. So I suppose, yes, of course, you never can know 100%, but considering the fact that so many people donate organs, for medicine and for dissection, wouldn't surprise me. So over here, this is another one. This is now a plant cross section. I just see that this is very red in color. So maybe I should try to, maybe there's a possibility somehow. I go auto exposure now. The auto exposure seems to be a little bit better here. I can also adjust the color temperature. See, I can change. This is I can do all of this over the over the control. Okay. So that's basically auto exposure target. Maybe that's a little bit. No, it's too dark. You get the idea. This is this is stained with carmine and malachite green. Okay, haha. Don't get me started. I just want to show you something. Just a second. I bought myself something, but I was not quite successful yet. I'm gonna get it carefully out of my cupboard. So it's basically I bought myself a stain called carmine, which is a red stain. And I've got it here. I just want to show it to you because why? Because this is actually the label actually says that this is also has been used also to stain this one here. This is a fern, cross section of the stem of a fern. Look at it; it's beautiful. You look at the cells and so on, and the colors are because it has been stained with two chemicals. One of them is carmine, which is actually a food coloring, and which is has been quite apparently not quite popular anymore as it used to be, but evidently produces very nice. Thank you. Lots of questions. I received again the questions. Thank you very much. I would like to just show you now what I bought here, and what I've been trying to do. As I say trying because my staining reactions with at least with my tissue cells from my cheeks and so on didn't quite work. But let's do the following. I'm going to move the microscope now carefully away. Now I really have to be careful. Let's move this down here. This is something that I bought. Yeah, one have to be really careful now. This is in powdered form, the stain carmine, and it is extremely red. Look at this, filled to the top, yeah, and even here on my finger now, extremely red, and it is as I said, it's sold as a food coloring, and what you have to do is you have to dilute it. Not diluted, you have to simply put it into some water, or also an acetate, acetic acid. Yeah? And then you can actually use it for staining chromosomes. And this is something I tried to do. Unfortunately, I was only partially successful. So I somehow maybe didn't get the concentration right, or there's some other issues. It's something that I'm still working on. There are some recipes online that actually say how you should mix it. I tried that, and unfortunately it did not dissolve very well, so I had to filter it as well. It was a little bit of a hassle, but the staining technique itself, I probably did not stain it long enough, the specimens, so I was not fully successful. So is the carmine pure, or has it added sugar? No, this should be actually pure. This is actually that's definitely pure, because it's was sold by a company for people who need this as a food coloring. And I know that there is food coloring that you can already buy ready mixed, so that you with sugar on it, and you can put it directly on the cake. But this is actually concentrated carmine powder, and it might not see it on my hands so much. But if you put some water on it, it really turns red. Yeah? So it's a very, very strong. It's a very strong pigment. But I have to still get the formulation right. Yeah. Try dissolving it in alcohol and then dilute it. The recipe is as follows. You're supposed to dilute it in about 30% or 50% acetic acid. Glacial acetic acid, and you should boil it to help it dissolve it, and then the remaining parts should be filtered away. Now, I, however, found out that the carmine dissolves much better in pure water than in acetate and acetic acid, so I'm probably going to dissolve it first in pure water, and then maybe I'm going to add the acid. A lot of recipes say that you should add acid because it improves the staining reaction. Boiling acid, what could go wrong? I tell you, this is pretty aggressive stuff. Okay, so this was 25% acid, and when you heat it up, you smell the fumes. Smell it. I mean, it's actually really very strong. Yeah, so you do have to be careful with this stuff. I'm not. Yeah, I'm just saying. Obviously. So let's have a look at this again under my other microscope. Oh my gosh. I cannot believe this. I'm going to flip the camera now. This scared me. Can I even do that? How can I flip the camera around? There is a bug sitting directly on my microscope objective. I have never had this before. And if you don't believe me, see this guy over here? So it is not focusing. There is, an insect on my objective. I've never had this before. You know what I'm gonna do now? This was not planned. Am I able to catch this guy? I'm gonna give it a try. I'm going to lower this here. This is obviously a bug and not a beetle. It's a Heteroptera, it's called. Get off, get off, get off. Where is it? Here it is. What a coincidence. Okay, go in here. Up, in it is. Here it is, I got it. So, now what? Now what? Let's try the following. Where is this? Just a second. Just a second. Go down. Here it is. Beautiful. Okay. Yeah. That's the one which was basically on my microscope objective, and of course some bio. I have to convert this immediately into a biology lesson. Yeah. If you actually look at the hind at the leg, down you go. If you look at this here, do you see those wings here? That the wings are basically membranous over here, so it's a bug and not a beetle. Yeah, so it belongs to the so-called Heteroptera. Yeah, and we've got a lot of these around. Okay, you know what? You get the idea. So let's put this one here again under the microscope. Gee. Okay, so that's basically the fern. Where's the fern? Nope. Here we go. And I need to focus because I lowered the microscope. At this stage, I mean, if I'm only able to. This is too bright. Here we go. And yeah. And that is essentially the red cell walls. That's basically because of the carmine stain. Yeah. So this is a stink bug. Yes. Where is it? It's now running around over here. I'm gonna carry it out later on. Yeah. Yeah. So yeah, that's basically the cross section of the fern. Yeah. What time is it? Yeah. What I'm gonna do now is I'm going to, because there is a whole page of questions that I got here, right? So what I'm gonna do is I'm just going to maybe talk about those here a little bit. Yeah. And yep. Okay. I'll just answer a couple of questions here. This is here. I was wondering if solutions of alcohol and water mean denatured alcohol and distilled water, or can I use isopropyl rubbing alcohol and tap water? You see, the point is the following. It really depends what you want to use it for. If you want to do standardized chemical reactions, then you always use distilled water and you always use pure alcohol. Generally, when you say alcohol, then you usually call it about ethyl alcohol. Otherwise, you just refer to it as isopropyl alcohol. It doesn't really matter for microscopy, because as soon as you mix alcohol and or water with a specimen, because the specimen makes it not pure anymore. You know what I mean? It's not, if you're concerned about distilled water, if you mix distilled water with a specimen, then it's not distilled anymore anyway, because of all of the substances from the specimens that actually go out into the distilled water. So it really doesn't make a difference, because in microscopy right now we're not working in analytical chemistry. So it doesn't make a difference. Yeah. Can you show us how to remove that back metal ring on the objectives? Yeah, I read this already before, and I don't know exactly what you're referring to. Maybe you are referring to, where is this again here? So maybe you're referring to this ring over here. This is actually something that either is, look, if this is the one, I don't know. Some of this can be simply taken out like this. That's it. Oh, I see that there is a threading in here as well. So actually, I should have probably rotated this. What you can do if you're interested in how an objective looks on the inside, in my other channel called Microbehunter Microscopy, if you go back in time, I think it was one of the first videos that I made several years ago. I actually took apart one of those objectives. Yeah, simply because I wanted to know how it looks like. So it's not in this channel, but in the other one called Microbehunter Microscopy, and you gotta go back all the way to the beginning. I think it was one of the first or second, I don't know, videos that I made. A very early video in any case, and I took apart one of these objectives simply because I wanted to know how it looks like. So you can try this as well. Does the colored ring only refer to the magnification of the objective? That's the next one. Yes, that's correct. It's simply an easy way so that you know what objective you have in place. And in my case, I've got yellow, red for 4 times, yellow for 10 times, green for 20 times, 40 times is blue, and 60 times is dark blue, and 100 times is white. So these are basically just the standards that they're using here. Yeah? How can we use it to study brain histology? I don't know exactly what you are specifically referring to, what the "it" is referring to. You mean microscopes? I can actually maybe go in here. Maybe I'm able to find the actual comment. Let's put it this way: if you wanna brain histology, I mean, I do have a couple of slides somewhere with cross sections of brain, not the human brain, but brain of certain animals. And if you actually look at this, then you're going to see, I don't know, thousands and thousands of tiny nerve cells. But light microscopes, you're not able to resolve how they are connected. You can see maybe the different layers, that there are different densities of nerve cells, but you're not able to see how they are connected or which brain cell is connected with another one. There are apparently imaging techniques out there that are able to do that have this high resolution, but the light microscopes that I have are not able to actually show which nerve cells are connected with which other nerve cells. The same question about denatured alcohol and distilled water. I always use denatured alcohol because it's the cheapest one, and I always use tap water. So if I want to, for example, remove alcohol for water, for example, from a specimen, I just show you. Maybe you remember from last time over here. These are the mushrooms. Right, and what I've done is I simply added concentrated denatured alcohol to those, and then what's going to happen is that now it's not concentrated anyway because the moisture of the fungi essentially went out into the alcohol and mixed with the alcohol, and the alcohol went in. So what you normally do is if you want to carefully dehydrate a specimen, is that you gradually increase the concentration of the alcohol. If you want to make it storable for a very long time, then you just basically exchange it again, make sure that there's very little water in there, and then you would simply use fresh alcohol again. I always use denatured alcohol, and I've got some plant specimens in here that I've been storing for over 20 years already, and they're totally fine in alcohol. So yeah, I would say from my experience, almost it can be stored almost actually indefinitely. So also note that pure alcohol, when exposed to air, will actually pull moisture from the air over time and will become less concentrated, well down to around 70%. Yeah, yes. First of all, that's actually one of the reasons why sometimes people use for the last stage isopropanol. If you take concentrated alcohol, then what is going to happen, and you open the bottle frequently, then the moisture of the air is going to reduce the concentration of the alcohol. But isopropanol will not do it to the same extent. So isopropanol is a so-called, under quotation marks, drier than the ethyl alcohol, denatured or not, doesn't matter, that we use. Yeah. So here. Yeah, so that's yeah. I recently saw your cheap telescope review, and he bought one for 25. Yeah, I saw many years ago, not a few years ago, I made a review of one of those cheap microscopes. Yeah. Seems like the 40 dry objective is the most finicky across different scopes. I would rather just go for 50 times and use oil, or stay at below 20 times. Generally, it's like the higher the magnification, the lower the depth of field. Every vibration, every bump is magnified immediately. So also for those microscopes here, because that's actually what I'm talking about today most, it works fine up to 10 times, but 40 times, light intensity is low already, depth of field is low. I don't have a condenser which bundles the light yet further. So yeah, you are indeed a little bit limited. With that, 40 times and above is already is more difficult to use. And some toy microscopes are, I don't know, they have these unrealistically high magnifications, over 1,200 times, and it's totally unrealistic. Yeah. So yeah. I tried a 60 times dry objective. It was all right, but it was on blood smear, so a super thin specimen. By the way, yes, I also have over here on my other microscope a 60 times. The distance, as a matter of fact, is so small between the objective and the specimen. The distance is so small that it can sometimes happen, or often happens, that actually the objective actually slightly touches the cover glass. So what happens is that when you focus, then you're actually able to see that the specimen is kind of pushed around as well, because of the objective pushing down on the cover glass. So the specimen has to be really extremely thin if you want to use a 60 times objective. Got to put on my glasses again here. Hi, Oliver. I'm following you also on Blue Sky. Okay, yeah. As I said before, I'm trying this out now. If it really keeps up to the promise that people say that, and then we're gonna see. Yeah, it's because I'm also cross-posting the links on Reddit as well. Yeah. So my best upgrade was a microscope camera. Many hobbyists drop out of the hobby for physical reasons. You sit more comfortably to look through the screen. Yes, this is also another thing, is people, especially when they become older, your eyesight goes down a little bit, and it's exhausting, especially if you've got microscopes only with one eyepiece. Yeah, kind of always looking through it with one eye is very exhausting. So if you connect a camera or even a mobile phone, that's also possible. You can try to connect a mobile phone and then connect the mobile phone to the computer so that you can use it like a webcam. Yeah, with it, there are certain apps that allow you to do that as well. Then this is more convenient. Yeah, and as a matter of fact, I do this very often. I quickly sometimes check through the eyepieces, but then when I'm doing any type of recording or filming or photography, it always goes over the computer. Yeah? It's so much easier and more convenient. Off topic, I want to start a paramecium culture. Yes, I'm going to talk about this, okay? Where in the pond do you have the best chance of finding them? On floating twigs or leaves, stones, or just in the mud on the bottom? Okay, Paul is asking this. This is an issue. Just want to share some experiences here. If you want to grow, I've got over here a jar as well, and of pond water and not successful. I will tell you why. So what I've done is the following. I went to the pond. I took some water, floating, just regular water, and I poured it back into the pond. And because of the turbulence, this swirled up some decomposing matter from the bottom of the pond, and that is what is scooped up. Yeah, so you can see that there is some algae, some whatever, in there. So I'm not collecting larger leaves or anything like that, but simply the smaller debris that has settled on the bottom of the pond. Yeah. So, and when you put that stuff under the microscope, you're going to find, especially during the warmer times of the year, you're going to find microorganisms, and if you're very lucky, you're going to find maybe occasionally one paramecium floating around, swimming around as well. So what you would like to do then is the following. Is you would like to then make a so-called an enrichment culture, and what you see in here is a wheat grain, oats cereal, so to say, that are dropped in here. This one over here was just a cereal, breakfast cereal, which was already pressed flat. But what I've also done is I've simply taken an oats and pressed flat between 2 spoons and dropped it in there as well. Could you use rice or cornflakes as well? Of course you could, but oats has the advantage that it kind of stays together and doesn't dissolve in the liquid. Yeah. And then what you do is after a couple of days, after 5-6 days, you take a tweezer, you take it out, and you put a drop of this oats on the microscope slide. And then chances are pretty good that you're going to find a lot of paramecia as well. And I've done that. And then after a couple of days, everything was gone again. Yeah. So this is a little bit of thing. Is what you have is you have a natural progression happening. Certain microorganisms start to grow rapidly, then they disappear again. And one of the problems is overfeeding. And the reason is because if you start to add food like cereals or whatever, then what you're doing is you're actually promoting the growth of bacteria. And then the paramecia, they feed on the bacteria. So it's a tiny food chain that you have. And that's basically, what you want to have. But if the bacteria are too many of them, then what's going to happen is that the oxygen concentration is going to drop too much, and then the paramecia are going to die off. Yeah. So you see, it's a delicate thing. And this pond water sample is now a couple of weeks old. I have not found any paramecia, so I don't know if there are still any left in here. So what I'd have to do is I have to go out to the pond again, collect a fresh sample, and start the enrichment process again. Yeah. So my suggestion is you just collect some stuff from the bottom of the pond. Do not use too much water, because they need a lot of oxygen, and basically if you fill it up too much water, then there is oxygen is not able to reach all the way on the bottom. And sometimes I've seen a lot of paramecia also growing around the green algae and green stuff, because they basically produce oxygen by photosynthesis, the algae, and it's where the paramecia also like to be. So I've seen that in different parts of the same jar, the concentration of paramecia can be vastly different. It was even that much different that I had different cereals and basically cereals on different places, and each cereal grain had different microorganisms on them. One of them was full with paramecia, the other one was full with, I think, rotifers, or I have no idea. Right. So this is something that you should also expect a little bit, that it's not always quite as reproducible, and there's some surprises as well. Yeah. But generally, highly recommended, because they're quite interesting to observe. Yeah. So so that's an Olympus that I'm using right now. I tried really hard to get a fetus hand slice, but it went. It was far too expensive. Yeah, I mean, in the medical field, they basically have a lot of these specimens. Yeah? I do find it interesting how often nice diatom slides go for so much more than a human. Yes, it's a question of collectors' items. Those diatom slides, as I already mentioned, maybe in some previous videos, back in the 19th century, what they've done is they physically placed the diatoms on the slide, and into nice patterns and arrangements. So basically, these were actually also artworks. Yeah. So I have to check a little bit what time it is. Okay, in a couple of minutes. Yeah, the monkey tail slide, my favorite one. Yeah, yeah. It's sad that the video did so poorly. This was one of the monkey tail video that I made. Just find it in the in this channel somewhere. It was one of my more unsuccessful videos. People didn't like it. Then I changed the thumbnail. I put a monkey on the thumbnail as well to make it less abstract. Yeah. I spent probably an hour talking with coworkers about the moral and ethical factors. Yeah, I already mentioned this. Yeah, that's indeed. There is, however, one thing that I just want to say here because it keeps on reappearing over and over again. A couple of years ago, when the YouTube channel was still quite young, I made a video about paramecium, which was the paramecium was crushed and squashed between the cover glass. It's not something that I deliberately did, but simply it happened, and I kind of was filming it. And people really complained about that because they had some ethical concerns. And what I can say is well, if you have ethical concerns, please have ethical concerns about the right things, under quotation marks. I'm not worried about microorganisms suffering because they do not have a nervous system. These are microbes. Yeah, they don't have a consciousness, right? So if you want to spend your ethical energy, then there are plenty of other things that really deserve more being ethically concerned about than a couple of microorganisms. I'm just saying this because some people really got upset with the video and flagged it and all sorts of things. Come on, it's just a paramecium being crushed. Right? It happens all the time. Yeah. So this is nothing to be concerned about because individuality at this level doesn't exist, like for the higher animals. Yeah? This concept of individuality or uniqueness or so, it does not apply to microorganisms. Yeah? Just saying. Okay. So I'm going to quickly go through some of maybe there are some more questions. Did you use Uprol liquid to mount insect or mites microscopic glass slides? Yeah, well, the slides that I showed you today, I did not mount. But when I made, for example, some slides some time ago of the Varroa mite, which is a parasite of bees. And for arthropods, I always use Uprol mounting medium because, well, it's the standard medium that entomologists use. And it has a clearing action. This means that even if the exoskeleton is very dark, after a couple of weeks, it really starts to clear up and become more and more transparent. Yeah. So for insects and spiders and arthropods in general, Uprol is a very good mounting medium. Also because you can take the insect directly from the alcohol, ethanol, rubbing or not, or isopropyl alcohol, doesn't matter, and you can directly transfer it then into the Uprol mounting medium, and it's still compatible. Yeah, this still works, right? So this is the reason. Yeah, let me. Yes, best entomology greetings from Ukraine. Okay, cool. I'm not an entomologist. I just have been doing a little bit with that. Yeah. So. You can you can wear an eye patch. Yeah, that's another thing. If you have only one eyepiece, then generally what they recommend is you should keep both eyes open and learn how to look through the microscope with only one eyepiece, but with both eyes open. It takes practice, but this also works. So. Can phase contrast objectives be used without the condenser analyst as a regular bright field objectives? So the question is if you have the short answer, yes. So the question is if you've got a phase contrast objective and you look at the phase contrast objective from behind, basically you take it out and then you're going to see a ring there, right? And this phase contrast, and this ring is the phase contrast ring, and basically, this ring will reduce the image quality a tiny bit, really not a lot. But you can use phase contrast objectives for regular bright field if you're specifically interested in that, because there is one question by by Timothy. Then I did make also in my other YouTube channel, in Microbehunter Microscopy, the other one, the more technical YouTube channel, I did compare a bright field and a phase contrast objective. Same make, everything the same, and I did a side-by-side comparison. Right? So if you're specifically interested in that, but the short answer is yes, you can use a phase contrast objective for bright field. There is a small loss in image quality though, because of this phase ring. Yeah. Okay, yes, I'm just skipping over it. Have you ever done a Vinogradsky column? No, I have not done this yet. For those of you who don't know what it is, this is a glass column that you fill up with sludge and so on, and then if you let it rest, you're going to see that there are different colors appearing, because in different zones you've got different bacteria growing depending on the oxygen requirement that they have. Yeah, so this is something I've not done yet, but it's of course maybe on a small scale, not even so difficult to realize. It's something that one might experiment as well. Yeah. So and then we're basically, I'm going up the comments. We're up at the stink bug again. Okay. So and what time is it now? Yeah, I think it's slowly time again to call it a day. If you have just joined next week, unfortunately for private reasons, I'm not around, and therefore I'm not able to do a live stream. However, what I'll do is I'll prepare a video that will be released at that time, not quite as long, and if it's a so-called a YouTube premiere. I've already done this before already when I was on holiday in summer, and in a YouTube premiere, it's like this. It will appear to be like a live stream, and there will be the comment section, and you can join in. But basically, I won't be there physically because it's pre-recorded. Yeah. So just for your info, in case you still want to join, and all of those videos anyway are available afterwards anyway in YouTube. Yeah. So that's pretty much all I want to say here. I hope I didn't forget anyone's questions or comments. Yeah, and I think I'm just going to leave it at that. And if you are not yet into microscopy, and if you're considering it, then I always recommend, just get yourself a not too expensive microscope at the beginning, so that the entry barrier is low, that you don't spend too much money at the beginning, and then if you like the hobby, you can always upgrade later on. Okay, that's a little bit my recommendation that I have, and I still have to check those. I checked, and unfortunately, if the water sample is too old, no paramecia. So take a fresh water sample as well. Last comment. I wish you all the best. Happy microbe hunting, and see you around, and maybe in this time again in 2 weeks. Okay, bye-bye.